Hey folks, Professor Finn here, and today we're going to be looking at the next chapter in our study of microbiology, looking at the book Pathology and Microbiology for Funeral Science students. Like, share, subscribe, get this video out, let's break the algorithm, and let's turn to chapter 21, Anatomy of Bacteria. Get the screen shared, get the pen up and running so that way I can scribble, and let's talk about anatomy of bacteria. So bacteria and other microorganisms differ substantially in their anatomical and physiological attributes from human cells. Binary fission, or simple transverse fission, is a method of asexual reproduction in bacteria in which the cell splits in two parts, each of which develops into a complete individual. This process can occur relatively quickly, producing multiple generations of a single bacterial cell within a brief period of time. A bacterial colony is a visible group of bacteria growing in a solid medium, presumably arising from a single microorganism. It requires approximately 1 million bacteria before a colony is visible without a microscope, and some bacteria are capable of reproducing once every 20 minutes under perfect conditions. Morphology. The morphology of bacteria refers to their size, shape, and arrangements. There are three basic shapes of bacteria with which we are concerned: spheres, rods, and spirals. There are three basic arrangements in which these shapes are found: pairs, chains, and clusters of grapes. While others are shaped like stars or squares, we will not be looking at those. Most bacteria are monomorphic, meaning they have only one shape, while others are pleomorphic, which means they have many shapes. Very important. Monomorphic, one shape, that one's easy. Pleomorphic, many shapes. Okay, one shape versus many. Cocci. Bacteria with a spherical shape include the word coccus in their names. The plural version is cocci. Spheres can be arranged in pairs called diplococci. Chains are referred to as streptococci, and a grape-like cluster is referred to as staphylococci. So two spheres together, diplococci, chains, long chains, streptococci, and then staphylococci means the spheres are arranged like clusters of grapes. These bacteria typically cause diseases like strep throat, food poisoning, scarlet fever, wound infection, and toxic shock syndrome. Bacillus. Some bacteria are shaped like a rod, and they include the word bacillus in this name. The plural version is bacilli or bacilli. Pairs of rods are called diplobacilli, and chains are called streptobacilli. Bacilli cause diseases like typhoid, diphtheria, and lockjaw. Spiral or helical shaped bacteria are named spirilla and resemble corkscrews. Bacteria that look like curved rods are called vibrios. Curved rods, vibrios, spirilla, corkscrews. Spirochetes are surrounded by an axial filament that allows them to control their movements by turning like corkscrews. Two important spirochetes are Treponina pallidum and Borrelia burgdorferi, which cause syphilis and Lyme disease, both of which are nasty. Cell walls versus cell membranes. Almost all prokaryotic cells have cell walls. The cell wall of a bacteria is a semi-rigid structure that provides the cell with its shape. The cell wall's primary purpose is to prevent bacteria from rupturing when the osmotic pressure inside the cell differs greatly from the osmotic pressure outside the cell. If there's a big difference in the concentration of materials, salts, and liquids in and outside of a cell, they will try to equalize. Okay? A cell wall basically says that, hey, if that happens, the cell's not going to crush in on itself, which is typically what happens if you don't have the wall, and if the change is too great, that also can happen. The body defends itself from certain bacteria by forming antibiotics that attack the cell wall. Some eukaryotic cells in plants, algae, and fungi have cell walls that are less complex and less rigid than those of prokaryotes, but most eukaryotic cells do not contain a cell wall. Not all bacteria have cell walls. The smallest known bacteria that can grow and reproduce outside the living host cell are mycoplasmas. You know that's important. Atypical pneumonia, the most common type of lung infection between the ages of 5 and 35, is caused by a bacterium known as Mycoplasma pneumoniae, and it does not have a cell wall. This is a problem if you are using a pharmaceutical agent that specifically attacks cell walls. It's like using a torpedo to sink an airplane, which sounds absolutely amazing in theory, but likely not to work too regularly. The cell membrane, plasma membrane, or cytoplasmic membrane, lines the inside of a cell wall of many bacteria. It is composed mostly as phospholipids with some proteins. Phospholipids are repeating units of fat and clusters of phosphorus. The cell membrane is selectively semi-permeable, which means that certain molecules and ions may pass through the membrane while others are restricted. A variety of antimicrobial agents are capable of killing bacterial cells by targeting the phospholipids in the cell membrane. Once the cell membrane is damaged, the contents of the bacterial cell are exposed and the cell dies. Cytoplasm. In eukaryotic cells, the term cytoplasm generally refers to the many functional organelles inside the cell. In contrast, the cytoplasm of prokaryotic cells does not contain functional organelles. It refers to the internal matrix of the material inside the cell membrane. Cytoplasm is about 80% water that is rich in enzymes, carbohydrates, and lipids, and it resembles a thick, cloudy, elastic fluid. The cytoplasm of prokaryotic cells contains a much higher concentration of inorganic ions than the cytoplasm of eukaryotic cells. Within the cytoplasm's nuclear area are both the bacterial chromosome and the plasmids. The nuclear area is not bound by a nuclear envelope, but contains the bacterial chromosome, which is a long, singular, and circular molecule of double-stranded DNA. The plasmids are small circular double-stranded DNA molecules that the bacterial cell can actually pass along to other bacterial cells. Plasmids are independent of the bacterial chromosome and can carry a variety of genetic materials that could produce toxins or increase resistance to certain antibiotics. Plasmid DNA is used for genetic engineering and cloning. Glycocalyx and capsules. Prokaryotic cells secrete a sticky, gelatinous coating called a glycocalyx that surrounds the cell wall. The outer coating on prokaryotic cells differs greatly between different species and is known as a capsule only when it is organized and firmly attached externally to the cell wall. If the glycocalyx is unorganized and only loosely attached to the cell wall, it is known as a slime layer. The capsule on some species of bacteria allows the cell to survive through adverse circumstances. In extreme circumstances, some bacteria can digest the sugars in its own capsule as a source of nutrients. Your book references capsulated versus uncapsulated in regards to pneumonia, and not surprisingly, the one without the wall easily gets its butt kicked by the immune system. Motility. Many bacterial cells also contain structures that allow them to move about, which is known as motility. Some bacterial cells have a flagella, a long whip-like filament containing appendage that propels the bacteria. Monotrichous bacteria have one flagellum. Amphitrichous bacteria have flagella on each end of the bacteria. Paratrichous bacteria have flagella distributed all over the entire cell. Do not confuse flagella with pili or cilia. Okay? Monotrichous 1, amphitrichous 1 on each end, paratrichous, it's covered in them. The flagella of prokaryotic bacterial cells function differently from the flagella of eukaryotic cells. Human sperm propel the cells through fluids with a wave-like motion of their flagella. Prokaryotic cells spin the flagellum at its point of attachment in either a clockwise or counterclockwise direction. Motility is a great advantage to certain bacteria because it allows them to move away from unfavorable environments toward a more favorable one. Some bacterial cells are covered with structures known as fimbriae and pili, which are shorter, thinner, and straighter than flagella. Neither of these provide motility. They only provide a means of attachment. That's important. Fimbriae and pili are not motile attachments. They only provide a means by which it can stick to something else. It can grab and hold. Pili are slightly longer than fimbriae. Fimbriae may occur at the ends of a cell or all over it. Pili are generally limited to one or two on a cell. You know that is going to be important. There's not a whole lot of pili, but fimbriae can occur at the ends or all over it. Gonorrhea is a disease caused by Neisseria gonorrhoeae. If the bacterium has fimbriae, it can attach to a host and cause the disease. If it does not have such methods of attachment, they cannot cause the disease. Endospores, or spores, are a thick-walled cell produced by a bacterium to enable it to survive unfavorable environmental conditions. The cell forms a highly resistant form of itself that can survive for thousands of years. An endospore is not part of the reproductive process because the number of bacteria is not increased by the presence of spores. The spore is a single bacteria, and it is not reproducing, it is protecting itself. When the endospore germinates, changing back into a fully developed bacteria from which it came, it is known as a vegetative bacterium. If the situation calls for it, the vegetative bacterium can form back into an endospore and the cycle continues. Under conditions of starvation, especially the lack of carbon and nitrogen sources, endospores may form within the bacterium. The process of forming an endospore is called sporulation. It is this ability to package its most essential components into a spore form that allows the bacteria to be extremely resistant. Think of it, all you preppers out there, you think of an endospore as your bug out bag. When things start getting tough, you grab the bug out bag and you jet. And what happens when things cool down? You refill the bug out bag and you prepare it for the next time. Endospores are resistant to antibiotics, most disinfectants, and physical agents such as radiation, boiling, and drying. Although endospores themselves are generally harmless until they germinate into a vegetative state, endospores are involved in the transmission of some pretty nasty diseases. Anthrax, tetanus, botulism, gas gangrene. Okay, so Bacillus anthracis, Clostridium tetani, Clostridium botulinum, Clostridium perfringens. Now, with Clostridium perfringens, careful note, if you've listened to the embalming lectures, this should be nothing new. Clostridium perfringens that occurs in a living person is known as gas gangrene. If it is occurring in a dead person, it is known as tissue gas. Endospores are extremely important to embalmers because they are resistant to disinfection. The primary difference between disinfection and sterilization is that endospores are destroyed during sterilization. Sterilization kills everything. Disinfection doesn't kill everything, it just lowers the number to a non-harmful level. Endospores also remain on countertops, embalming tables, embalming instruments, and damn near anything else they're on unless they're sterilized or they're killed with a sporicide. For this and multiple other reasons, we always wear our PPEs and practice universal precautions, and we shouldn't be just chilling in the prep room to hang out. Microscopy. Since bacteria have unique characteristics such as different forms of motility and different structures, the scientist must first determine which structure should be observed and then utilize the appropriate technique to see them with the microscope. If the appropriate technique is not chosen, the bacterial structures appear transparent and motile bacteria would have to be motionless to be visible. There are two basic techniques used to observe the morphology of microorganisms through the microscope. A living specimen, which can be suspended in some fluid, or smears of a specimen that can be dried, fixed, and stained on a piece of glass called a slide. Fluid suspensions. If the scientist wants to observe the structures of microorganisms while they are alive and to see them move, the specimen is suspended in fluid. And there are two methods, the hanging drop technique or the wet mount technique. Both of these techniques are used to observe the morphology of spiral bacteria, to observe the movement of bacteria, to observe cell inclusion bodies like fatty materials, or to observe cellular reproduction. The wet mount technique begins with a ring of petroleum jelly on the cover slip, and then a drop of the specimen is added to the center of the cover slip. Then a glass slide is placed on the cover slip, and the two are gently pressed together so they are sealed. When the slide is turned over, it can be placed on the microscope, and the specimen can be observed through the cover slip. The petroleum jelly prevents airflow from disturbing the microorganisms. The wet mount technique is best suited to the observation of larger microbes such as fungi, protozoans, and yeasts. You know that's important. When suspended in fluid, both motile and non-motile bacteria exhibit Brownian movement. Brownian movement is a rapid motion due to the movement of the liquid's molecules around the bacteria. Motile bacteria may dash about, spin, or move in a wave-like fashion. To observe, now we're on to smearing. To observe the morphology of microorganisms, a thin layer of bacteria called a smear is typically prepared on a glass slide. A smear is prepared by using a special technique to place bacteria from a bacterial colony culture onto the slide with an inoculating loop, which is a loop of wire affixed to a handle. The specimen is allowed to dry on the slide. The bacteria is fixed to the slide by heating it so that the proteins in the bacteria coagulate and stick to the slide. The bacteria that have been fixed to the slide are not dead. The fixing simply prevents them from falling off the slide as it is handled. After drying and fixing the smear, it is typically stained so that the desired structures may be visualized within the microscope. A common dye to observe DNA or RNA in a cell is methylene blue. Both methylene blue and crystal violet are classified as basic dyes because the dye's ions have a positive charge and stain alkaline structures in the cell. Acidic dyes, such as acid fuchsin, Congo red, and eosin, have a negative charge on the dye ion and are used to stain positively charged cellular proteins. Fats in cells are observable with a stain called Sudan black. All of the simple stains are designed to sustain the entire cell so that its structure can be seen more clearly, while differential staining is far more complex because multiple stains are employed to stain certain parts of the cell. Dr. Hans Christian Gram developed his method of staining in 1884 while working in a morgue. Gram staining is one of the most important and widely used methods of classifying bacteria into two groups, gram-positive and gram-negative. Gram staining is a four-part process. A purple dye is added to the bacteria fixed on a slide, and all bacteria that are able to absorb this dye are stained purple. A dilute solution of iodine is added, creating a less soluble dye-iodine complex. An organic solvent, such as ethanol, is used to remove the purple dye-iodine complex from some, but not all of the bacteria, depending on the genus of the bacteria. And a red dye such as safranin is then applied, staining all the bacteria. Bacteria that appear red had the dye-iodine complex removed during the process and are called gram-negative. Bacteria that appear purple due to the masking of the red dye by the purple dye are considered gram-positive. All gram-positive and negative means is some turn red and some turn purple. The ones that are purple, the dye stuck around. The ones that turned red, the dye left. Gram-positive bacteria have cell walls that are composed of few lipids, while gram-negative bacteria contain a high concentration of lipids in their cell walls. Professor Finn, for the love of God, why is this important? Well, matter of public health. If it is purple without a lot of fats, breaking that wall is going to take different techniques than if it does have fats in the cell wall, because then we can attack the fats and the cell wall will crumble. Gram-positive bacteria are more permeable to basic dyes, while gram-negative bacteria are less permeable to them. Gram-positive bacteria tend to have more complex nutritional requirements than gram-negative. So starving them takes a different type of solution, or trying to starve them will require different solutions. The more complex, the easier it is to kill. Basically, the more complex, the harder it is to kill because of the fact you can't just starve it of one thing, you got to starve it of a bunch. If it's one thing, you just take the one thing away from it and it dies and have a nice life. Gram-positive bacteria are generally more resistant to physical treatments and or killed easily by penicillin and sulfonamide drugs, while gram-negative bacteria are more susceptible to streptomycin and tetracycline. The proper staining method must be chosen to observe the desired structures on both cells. The correct choice of stain is an important part of the examination of any specimen, as your book states in the concluding paragraph to this chapter. So folks, that concludes this lecture. We'll see you next time. Once again, like, share, subscribe, share this with a friend. Drop something in the comments below to let me know I'm doing a good job or tell me where you're from. And we will see you next video.